It's definitely a pleasure to be in front of all of you since I began my career to where I got to this point because I loved my high school biology course so much. So I certainly hope that that can continue to help some of you in some way. So today I'll focus mostly on the idea of how we're using mouse models and stem cell biology to better understand the human disease lung cancer. Just to give you a brief outline of today, I'll speak to you a little bit about what's known about the impact of lung cancer in the clinic. I will cover the different types of cells that exist within the mammalian lung, and I'll touch very briefly on lung development. Then we'll get into how we actually were able to uncover a population of stem cells in the mouse lung and how we can use mouse models of lung cancer to combine these themes to begin to understand the basic biology of these tumors. So many of us don't often think about lung cancer, but it is actually the most prominent cause of death from cancer in the world. And even though we often hear much more in the news about prostate and breast cancer in men and women respectively, lung and bronchus tumors are actually the second most commonly diagnosed tumors. However, lung tumors in both men and women are the #1 cause of death. And the statistics shown here are those that are expected this year from the United States alone. So here, as many as 160,000 people in the US are expected to lose their battle with lung cancer this year. And this is not a problem of the United States alone and occurs throughout the world. Now, we all know that the majority of lung cancer cases are associated with tobacco use. However, I'm using this slide to show you that statistics have certainly seen a decrease in the incidence of lung cancer here in males by 2003. And perhaps this may be due to the increase certainly in the use of cigarettes and the knowledge of their link to lung cancer. However, in certain populations, for example here in women, at least at the time of this, a decline in lung cancer had not yet been seen. In fact, in women there is an increased incidence of lung cancer in those who have never smoked. And even though we can certainly, it's very important to spread the word about preventing tobacco use, there are still millions of people in the world who perhaps have already undergone the changes that will make lung cancer start in them. And so it's certainly a disease we need to continue to think about. And aside from the incidents associated with tobacco, environmental insults and other issues that may also lead to lung cancer formation are certainly not on the decline, at least at this point. So the devastation of lung cancer and the fact that it is such a mortal disease is probably due to at least two major reasons. And one is that the majority of lung cancer cases, shown here about 50%, are diagnosed when there is already a distant metastasis, where the tumor has gone on to either a lymph node nearby or actually a distant metastasis. And unfortunately, those cases have about a 2% 5-year survival rate. So most lung cancers are being detected when it's already too late. Now, the statistics here do give us some hope in that when a tumor is found at an early stage, there is a much greater 5-year survival rate. So these 2 points, I think, give us really point to what we need to focus on in lung cancer research. First, we can really link these 2 bars by saying if we can find better ways to detect these tumors at an earlier stage, it would certainly make a great impact on the survival for lung cancer patients. And because so many people today may, in the coming years, develop lung cancer that we haven't been able to detect, we need more effective treatments for the advanced diseases. And so my research has taken these 2 goals in mind. And the approach that we've taken is that in order to find these tumors earlier, we need to understand which cells in the lung are first involved in the process of lung cancer formation. And we also need to understand the biology of the advanced tumor which cells in the tumor really make it continue to grow and metastasize, and how can we target those important cells more specifically. So these are the things that we hope to achieve.
Can you tell us what the difference is between localized and regional? I'm not familiar with that.
So, and I should maybe try to get you some more information on that, but my understanding is that the localized disease would be lymph nodes that are very close to the lung. Now, what exactly regional means, I apologize for putting it on here and not being able to tell you specifically, but I think it's, with lung cancer, there are actually numerous stages, and the farther away the metastasis is within a lymph node, which is usually what's sampled to detect the metastasis, so the regional is probably some threshold that the physicians have set, that it's sort of close to the lung, but still not as far as distant. Distant would probably be more like in the liver, in the bone marrow, and those kinds of other organs. So with the goal in mind of understanding the biology of lung cancer, we really need to understand the biology of the lung. And we can think of the lung as actually many different organs or niches. And this word, which you may have heard yesterday, refers to the microenvironment or the region in which a particular set of cells or one cell interact with the other cells around them and other important molecules. And in the lung, we might think of many different of these organs or niches, and we can start with the trachea, and as we go down into the bronchi that's split here, and then the bronchioles, these make up the conducting portion or the proximal airway, and so the air would be passing in this manner. And then we get out into the terminal bronchioles, which are the last part of the conducting airway, and then into the alveolar spaces and the alveolar sacs, which many of us might be more familiar with, where the gas exchange actually occurs. Now, importantly, there are these different locations, and the cells that line these structures have a very different shape and function. So up in the proximal portions, we can find cells that look like this under the microscope, where they have a very complex structure and ciliated pattern, and I'll show you more detail in the next slide. In addition, when we get further into the bronchioles, there's a more simplified or smooth epithelial layer, and down here in the alveolar space, the cells become more flattened. Now, to make this a little bit more complex, I certainly won't expect you to remember all of these cell types, but if instead of traveling down the airway in this fashion, if we take a cross-section in each of these different regions, and we look at this cartoon, you can see that in the up in the trachea and the upper airway, there's what's referred to as a pseudostratified layer of cells. And many of these are different types of epithelial cells, like the ciliated cells. There are goblet cells, submucosal glands, and even these very tiny basal cells. And as we get farther down into the lung, it becomes more of a single cell layer, which contains Clara cells, which are the non-ciliated bronchiolar cells, and you'll hear a lot more about these cells in the talk, as well as ciliated cells. And then in the alveolar space, we find the alveolar type 1 cell, or often referred to as the pneumocyte. This is actually the cell that performs gas exchange, and the alveolar type 2 cell, which is a very cuboidal cell that secretes surfactant, and the surfactant proteins are very important for creating surface tension, which allows gas exchange to occur. So you can see that there is certainly a diversity of cell types within the lung, and in addition, luckily we have a certain number of molecular markers that we can use in order to identify these cell types in our studies. So again, it's not important for you to remember the names of these, however, just to know that we do know, for example, that FOXJ1 is a marker for ciliated cells, and it's staining here in green. P63 is a protein that's found in the basal cells in that upper airway. A cell type that I didn't mention was the neuroendocrine cell, which is still quite a puzzling cell type and has markers such as calcitone gene-related peptides shown here in red. And in addition, in the bronchioles, we have those Clara cells that we can stain with SCGB1A1, or what I'll refer to it as Clara cell antigen It's also known as Clara cell secretory protein. And in the alveolar space, we can mark the type 2 cells with surfactant protein C. So even though we have this list, this is actually a short list for molecular biologists to work with, so something I'll mention is we certainly like to have more than 1 or 2 markers for each of these cell types and better understand their function. Now, you heard, I assume, from Dr. Rajagopal yesterday very nicely about lung development, so I won't cover that in any detail here. But just to remind you that the lung does develop from the endodermal tissues, the same tissues that would give rise to the intestine, the pancreas. The lung also starts as a bud from that endodermal tube. And the development that occurs for the lung within the mouse actually very closely mimics human lung development. There are certainly differences, but because of their similarities, we can study both lung diseases and lung development in mice and use that to understand human conditions. And the process of lung development occurs through a number of different stages that even continue a bit into after birth. And this process is known as branching morphogenesis, where it becomes increasingly more complex through the different stages with the number of these branches that eventually make these alveolar sacs where gas exchange occurs. Now, interestingly, this branching pattern of development also is very similar in the fruit fly. And so in the fly, we find the tracheal system, which is outlined here in yellow, including some epithelial cells as well as the vasculature, both shown in yellow and red. And because of the similarity of the very early stages of development, it has been possible to use genetics in the fly and uncover some of the genes that are required for lung development, including for these processes of making these different branches. And again, I won't get into it in detail, but if we have time at the end of the talk, I'll just mention some of these genes listed here, including fibroblast growth factor, or sprouty, or sonic hedgehog, which have been implicated, shown to be required for lung development in Drosophila, tracheal development, but also are important in lung development in mice and humans, and interestingly, now have in many cases been linked to lung cancer as well. So as we consider the biology of the lung, I also want to remind you that I've shown you more about the epithelial cell types that exist, but these cells are also intimately associated with fibroblasts, smooth muscle cells, as well as the vasculature. And so it's a very complex organ, and this has actually made it very difficult, for the most part, to study stem cell or progenitor cell biology in the lung and other solid tissues. And in fact, for many years, the major way of studying lung regeneration has been done in rodents, and looking at the response of rodent cells to lung injury models, mostly with chemicals. And these studies, which I'll show you a few examples of, have led to the hypothesis that each of these regions within the lung might harbor their own stem or progenitor cell population. And the idea is that, for example, within the trachea, that there may be a distinct stem or progenitor cell that will respond to injury only in this location, but that yet another separate type of stem cell exists down near the alveolar space to respond to injury in that location. But actually, many of these studies have happened for years, but until some of the work I'll show you, it actually wasn't possible to isolate these putative stem cells and show that they have stem cell function. So some of the rodent models of lung injury that have been used include damage to the Clara cells. And these are the cells that here are stained with this antibody shown here in green to detect the Clara cell marker. And again, these are the cells that are lining the airways. And a model of lung injury, which I don't have, oh, here we go, naphthalene, is a chemical that we can inject into the mice, and Clara cells will specifically be ablated by naphthalene. And here you can see a normal lung with many Clara cells, and this lung on the right has been treated with naphthalene, and most of the Clara cells have disappeared. However, if we look at the green staining, the Clara cell marker, there are cells here at the very tips of the terminal bronchial that seem to be resistant to naphthalene. And there are also some, which may be hard for you to appreciate in this tiny image, cells here near the red staining cells that are also resistant. And the red cells here are those neuroendocrine cells that I mentioned. And so there seem to be two regions where naphthalene-resistant cells reside, either near neuroendocrine cells or here at the end of the terminal bronchial. Now, this area is also referred to as the bronchial alveolar duct junction. That's because this is the last portion of the bronchial before it opens up into the alveolar space. Now, the fact that these two cell types are resistant to damage, and that later in a normal mouse, you can essentially go from this image to the one on the left, in that normal mice can repair this kind of damage, has led to the idea that these resistant cells might be stem or progenitor cells that go on to give rise to new cells that will line the airway. And so this is some of the first work that really showed us the regions where these stem cells might reside. So just to give you another example of that, there are actually numerous types of damaging agents that people can use in rodent lung. And for example, I've just shown you naphthalene, where some of the most Clara cells will die from naphthalene treatment. However, there are these variant Clara cells that are resistant, and they reside in the terminal bronchial. Now, another type of injury is bleomycin. This is actually an antibiotic that is used commonly in treating some types of cancer. And it also damages the pneumocytes, the alveolar type 1 cell. And other studies have shown that the type 2 cell, this cuboidal cell, is resistant to bleomycin and can be seen to divide after the death of the type 1 cell. And so it's been hypothesized in the literature for many years that alveolar type 2 cells might be a stem or progenitor cell in the lung. Now, aside from these kinds of studies, however, there's actually been very little genetics or cell isolation that back up the idea that these cell types are stem or progenitor cells. So it's certainly something that we still need to improve on within the lung. And actually, it's thought that the basal cells might be a stem cell in the upper part of the lung. And the work that I'll show you, however, will point to what we think we've uncovered as a stem cell. And so the goal here would then be to define what these different putative stem cells might be and actually show their stem cell function. Now, at this point, I want to remind you then how we can actually demonstrate stem cell function. And as it's important to keep in mind the defining features that make stem cells unique from other cells in the body. So stem cells are the unspecialized cells that can give rise to the differentiated or mature cells that function within a tissue. And at the same time, they are unique in their ability to self-renew, which means that they can self-maintain and keep giving rise to more unspecialized cells. And this means that they have a proliferative capacity, or they're able to divide many times, perhaps over the lifetime of an organism. Now, some stem cells might also only exist during a particular stage of development. So the requirement for this to be present throughout lifetime is certainly not an ultimate requirement for defining a stem cell. Now, progenitor cells, in contrast, can still give rise to more specialized cells, but they don't have the ability to self-renew. And then it's thought that the differentiated cells are often post-mitotic in that they are not able to divide and yet carry out the function of a tissue. So as we keep these defining features of self-renewal and differentiation in mind, we then want to apply this concept to the lung. And a nice model for doing so has been to look at what is known about the hematopoietic system. And hematopoietic stem cells have actually been very nicely defined. And it's already known that there are hematopoietic stem cells that are referred to as long-term as well as short-term. They have a more limited ability, but these cells can give rise to progenitors, which ultimately give rise to all the different specialized cell types within the blood. And because we can actually draw this very nice lineage map, the blood is really a wonderful system to look at and learn how have they learned about stem cell biology. Well, one important aspect is that it has been possible to identify markers which are on the surface of most of these cell types and to isolate cells based on those surface proteins specifically and get very pure populations of these cells. And because of that, it has also then been possible to transplant different types of cells into the bone marrow and show, for example, that the hematopoietic stem cell could give rise to these specialized cells. Another technology that has made this possible is the ability to sort cells based on those surface markers. So we certainly would like to get a similar understanding of this kind of lineage map in the lung. However, we're really not there yet, but we're just beginning to be able to do this. So with keeping this goal in mind, I went on to begin to look more carefully at the known markers that we had for the more specialized lung cells. And this was work that I did during my postdoc, and I simply took the mouse lung normal tissue sections and performed immunofluorescence and stained for these proteins shown here. So CCA is the Clara cell marker, which you can see stains in red here, staining the Clara cells which line this terminal bronchial. And then the green is SPC, this punctate stain, which is a marker for the alveolar type 2 cells out here in the alveolar space. But what I found was that there were individual cells that actually had both of these proteins. And this was a quite novel finding because previously it was thought that these were cell type specific markers and that in the adult lung, cells would only have one of these or the other. Now, the other interesting thing was that these cells were very rare, and yet they always occurred in one location when we were able to find them, and that was right here at this bronchoalveolar ductal junction. And you might remember that a few slides ago I told you that this is where cells reside that are resistant to the naphthalene treatment. And so this region had already been proposed to be a lung stem cell niche. However, no one had isolated the cells from that niche. So that was very intriguing to us. And in addition, studies in lung development had suggested that in the embryo, there might be cells in the lung that were positive for both of these markers, and that those cells would go on to give rise to the more specialized cell types. Now that hasn't been confirmed and still needs to be performed, but that led me to ask whether or not these cells, because they had the two markers of two different types of specialized lung cells, might they perhaps be able to function as a stem or progenitor cell to give rise to either type. Another intriguing thing about these cells and where they resided is that in a mouse model of lung cancer that I'll describe for you, we saw that the earliest forms of tumor seemed to grow right in this area of the lung. And so it was very interesting to us that these cells might somehow be involved in the process of lung cancer formation. Now, these two markers, which I didn't precisely point out to you, are staining the cytoplasm of these cells, but they're not on the surface, they're within the cytoplasm. And so having these intracellular markers is useful for staining tissue sections, but it does not allow us to isolate cells when they're still alive and then grow and culture these cells and study them further. So we needed a way to take these cells out of the lung, and I went on to find that we could do so using a different set of protein markers that were on the surface of these cells. And what we did was to test for cell surface proteins such as SCA1, which were known to be on hematopoietic stem cells, and at the time it was thought that this protein might be on other types of stem cells in other adult tissues. And so I isolated cells from the mouse lung, where we start with the lung, we do an enzymatic treatment to get single cell suspension, and then we incubate those cells with antibodies that are conjugated to a fluorescent color and will detect this cell surface protein. And this is referred to as fluorescent activated cell sorting, or FACS. And each dot on this plot represents a single cell. So what you can see are the majority of cells are up here, where this is just indicating the density. And that means that in our lung cell isolations, the majority of cells here are positive for the marker CD45, which is a marker of the hematopoietic cells, or CD31, which is an endothelial cell marker, a blood vessel marker. So that means that when we isolated these in our cell prep, most of the cells were hematopoietic or endothelial. But if we exclude those cells and only focus on the cells that are positive for SCA1 and at the same time negative for the other markers, we can actually use FACS to sort out and only obtain these cells in a new tube, and then take those cells and stain them for the other cell markers that I described for you. And when we did that, shown down here, and I apologize, you might not be able to see the contrast very well, we found that most of the cells in this region were positive for both CCA, which should be showing up a little bit in red here, as well as the SPC in green. And we also found that ciliated cells were also in this group, and here they're just taking on that red stain for CCA. We also found that if we take the cells that are positive for SCA1 and another marker, CD34, that we can even better purify for the cells with the two long markers. And here we never found ciliated cells, and we only found these cells that were positive for both CCA and SPC. So this essentially gave us a great tool with which we could isolate these cells, because now staining here, these cells we had to fix and therefore kill in order to show that they had these markers. But when taken directly from the fax machine here, these cells are still alive, and so we could put them into culture and test their properties. And we did that, and through a number of studies that I haven't outlined here, but I'd be happy to answer any additional questions, we were able to show that those isolated cells had stem cell properties. And because of that, we decided to call them bronchial alveolar stem cells, or BASCs. So first we were able to show that they could self-renew. And we did that by performing a limiting dilution analysis. And that was done by plating different numbers of those cells, either what I'm labeling here as BASC is that SCA1 positive population, or the total unsorted lung cells, or what we were able to isolate as a mostly pure population of alveolar type 2 cells. And we then took those cells from the fax machine and plated them onto 96 well plates, and this was on top of cells called feeders, which are mouse embryonic fibroblasts that are also used to grow embryonic stem cells. And these feeder cells provide a supportive layer underneath the epithelial cells that we're plating them on. And when we do that, we can see epithelial colonies form. And what this graph is then indicating to you is the percentage of wells in our plate that did not form colonies. And so this more steep line we observe here means that this population was much better at forming colonies than the total lung, which I showed you is mostly hematopoietic cells, so that's not surprising, but even better than the alveolar type 2 cells. Now, the important thing was that not only could they form colonies to begin with, but we could take those colonies and remove them from the plate and get a single cell suspension of them, and then again, plate different numbers of these cells, and that these cells were able to form secondary colonies, and we could then do the same thing over and over again, and showing you here up to 9 times, but we've been able to do this up to at least 12 times, and we're still working with these cells. And this indicates that they are able to continue to proliferate multiple times, and therefore they're able to self-renew. And in contrast, the alveolar type 2 cells were able to form colonies, although still much worse than BASC to begin with. But also, when we took those colonies and replated them, they never formed secondary colonies. So even though I mentioned earlier that for many years the type 2 cells have been referred to as a stem cell, that at least in this culture assay they cannot renew, which would suggest they're more likely to be progenitors or differentiated cells. Now, this was to demonstrate that these cells could self-renew, and at the same time we could take these cells and put them on a different substrate called Matrigel, which contains extracellular matrix proteins and growth factors. And what we found is that even though we started with cells that here are positive for the 2 lung cell markers, that when we put them in these conditions, they were able to give rise to cells that were only positive for SPC, the alveolar cell marker, or only positive for CCA, the bronchiolar cell marker. And this suggests to us that these cells are differentiating in this condition. And we could have them do this whether they were at the 5th passage or the 1st passage or so on, which suggests that while they're able to continue to renew, they can still give rise to differentiated cells. And that's why we decided that we had shown that they have stem cell properties and that we could call them VASCs. And because they could give rise to both alveolar and bronchiolar cells, we hypothesized that these are a stem cell population in the distal lung that can maintain both the bronchiolar and the alveolar cell types. So to begin to ask that question in more detail, we then said, well, if we can observe these phenotypes in culture, we should then be able to see these cells respond to injury of these cell types in vivo. And so we used that model of lung injury that I described for you earlier, naphthalene, and we injected mice with naphthalene and observed these cells at different time points after injury. So just to remind you, in red are the Clara cells, and the VASCs are here at the arrows at this junction. And in this case, the naphthalene-treated lung at an early time point, you can see, is almost completely stripped of these Clara cells, and some of them are still getting sloughed off. And what we did was to quantify the number of VASCs at the end of the terminal bronchioles, TB here, and we found that at different time points after injury, the number of VASCs was never decreased, and in fact, at 1 week after injury, there was an increase in the number of these cells. And in other studies, we found that VASCs, when they're in normal lung, were never dividing, or did not seem to be actively in the cell cycle. However, after naphthalene injury, they did divide, and this probably leads to the increase in the number of these cells that we saw. So these data suggest to us that VASCs are the first cells in the terminal bronchiole that can respond to injury of the Clara cells. So at the very beginning of the talk, I showed you this picture of pulmonary cell diversity, but what was lacking in this picture is the VASCs, which we've now defined. And so we've come up with this new cartoon to show you the cell type that we've identified, VASCs, which are depicted here in yellow. And they reside at the end of the terminal bronchiole. And I've shown you that in vivo they can respond to injury of the Clara cells, shown here in red. And in other work, we've also seen that they can also increase in number in response to alveolar type 1 cells from bleomycin, as I referred to earlier. And so we think that these cells can self-renew and give rise to the specialized cell types in this region of the lung if injury occurs. And we also now have a few markers which we can study these cells with in more detail. We know that they're positive for the Clara cell marker, CCA. They're positive for SPC, whereas in contrast, alveolar type 2 cells are only positive for SPC. We also have cell surface markers that we can isolate these cells with. Now, even though we have a few markers, one of the goals of my laboratory's research will be to add to these lists in order to make it possible for us to further study these cells. And in addition, we'll work on creating tools where we could ablate certain genes, for example, only in one cell type or another, and determine if they really are the important cells that we think they are. So now that we've covered the diversity of the normal lung and how we discovered a population of lung stem cells, I also want to remind you that that lung tumors are also come in a number of different forms. And if we look at the pathology of lung cancers as they occur in patients, we can find a number of different morphologies. And interestingly, these different types of lung cancer very frequently resemble the cells in the normal lung where the tumors first seem to grow. So the most common type of lung cancer in the US and mostly in the world is non-small cell lung cancer. And this name came from the fact that these cells are very different from the other major type of lung cancer, small cell. So small cell was named because the cells appear small under the microscope. And it's actually thought that these might come from a neuroendocrine lineage. And they behave very differently in that there are different courses of treatment for small cell lung cancer versus non-small cell. So any tumor that does not have this appearance would then be non-small cell. And the most common form of this is adenocarcinoma, which is shown here. And in many cases, this is a very aggressive disease that becomes invasive and this is actually the most common type of lung cancer in the United States. There are other cell types like squamous cell, as well as large cell, and these are very poorly understood. And in addition, bronchial alveolar carcinoma also resembles adenocarcinoma, but is usually a more benign form of the disease for the most part. Now, interestingly, these tumor types often resemble the bronchial or alveolar cells. Now, because of the diversity of lung tumors, it will become obvious to you that lung cancer is really a number of different diseases, and we probably need to understand each of these diseases separately. They may have very different responses and origins. And this kind of molecular data actually goes along with that statement in that in patient samples for lung cancer, if we look at genes that are known to be either tumor suppressors or oncogenes, we know quite a bit about the different types of mutations in human lung cancers. And for example, some genes like P53 are very commonly mutated in both non-small cell lung cancer and small cell. However, interestingly, there are also differences. For example, the RAS oncogene is much more frequently mutated in non-small cell, whereas the retinoblastoma gene, RB, is much more commonly mutated in small cell lung cancer. So this then suggests that there are probably a diversity of cell types that might be particularly susceptible to a certain mutation, and somehow this combination of cell type and mutation leads to a particular type of lung cancer. And we really don't understand this process at all. So what has been done is to use many different methods for modeling lung cancer in the mouse, because, as I mentioned very early in the talk, that most human lung cancers are diagnosed as a later stage, and therefore it's very difficult to understand the earlier processes there in patient samples. And so in the very early days, most people were using chemicals to induce cancers of the lung in mice, and more currently we're moving towards using genetically engineered models that more precisely mimic the human disease. So the mouse model that I've used for my studies was previously created in my postdoctoral mentor's lab, Tyler Jacks, and this model is referred to as a conditional allele of oncogenic KRAS. And this is one of the genetically engineered models of lung cancer. And Dave Tuvison in Tyler's lab previously made this mouse strain, in which he was able to engineer a mutation in the KRAS gene. And this mutation at codon 12 is found in about 30 to 50% of all of human lung adenocarcinomas. And it's also found in a large percentage of pancreatic cancers and other tumors. And so what he did was to engineer this mutation such that it was not normally expressed, and that was because he put this transcriptional stop element in front of the mutation. And this stop element was flanked by special sequences called LOXP DNA sequences. And these LOXP sites or sequences are not normally found in the mammalian genome, but can be engineered here. And the way that this system works then is that every cell in this mouse is carrying one normal copy of the KRAS gene and one of these mutated copies that is not being expressed. However, when we expose the cells to the Cre recombinase protein, the Cre recombinase recognizes these LOXP sites and deletes the stop element. And then this then allows for the expression of the oncogenic or mutated form of KRAS. And the way that we use this allele in order to engineer lung cancer is that we prepare a mixture of adenovirus that is expressing this Cre recombinase, and the mice inhale this virus, and therefore we're only exposing the lung to Cre, and therefore activation of oncogenic KRAS. So this makes it a very tissue-specific expression of an oncogene, and it actually represents the same amount of expression that you would find in the normal tissue. It also allows us to know precisely when we started the process of the tumorigenesis, and therefore we can study the very early events. And so what Erica in Tyler's lab showed was that at very early time points after the Cree inhalation, that the mice had hyperplasia. And these are all hematoxylin and eosin stained sections of lung tissue from these mice. And this is a hyperplasia, or AAH, right here, where there is a thickening or an increase apparently in the number of type 2 cells. And this type of lesion has been seen in many patients, and it's been proposed to be a precursor to lung cancer in humans. Now, at early points, we only saw this lesion, but by 6 weeks after Cree, we found these adenomas, which are very self-contained and still have fairly normal-looking cells. But by 16 weeks, these cells are very atypical, and the tumors have become advanced, and these are adenocarcinomas. Now, in other studies, the Erica Jackson, who was in Tyler's lab, went on to show that if she combined this KRAS mutation with loss of the p53 tumor suppressor, that these tumors become even more aggressive, and these are much more similar to the types of human adenocarcinomas that are seen in patients. So I used this system because I knew that at this very early time point, we started the process of KRAS activation, and then we could see which cell types might be involved in this very early process. And before we get to that, I don't want to overwhelm you, but this was just to point out that a number of different mouse models of lung cancer have been created using similar approaches, and in some cases these tumors have neuroendocrine features, but most of them are adenomas that look very similar to the type of cancer I showed you. And we still need to create more mouse models of the other types of lung cancer like squamous and large cell. There has been a mouse model of small cell lung cancer that has been developed, and this is using a combination of deleting RB and p53, which I mentioned to you is one of the genes most commonly mutated in small cell lung cancer. So with all of these tools in hand, we can then combine the knowledge of these tools with our knowledge of stem cells and begin to ask, how might these ideas be connected? And in fact, when I first started my postdoc, the idea of how stem cells and cancer might be related to one another. So many of the properties that most cancer cells seem to share is that they can divide many times and essentially without limit. And this might be similar to the way stem cells are able to self-renew. And also a tumor is very heterogeneous, and if it does start with one or a few cell types, then this process might be similar to differentiation. And so we'd like to understand if there is a connection. And this is along with the idea that, as I mentioned to you, that perhaps because stem cells have these unique properties of self-renewal and differentiation, that it's possible that they might be the cells that are more likely than any other cell to give rise to a tumor. On the other hand, a progenitor cell is still able to divide, and perhaps if a progenitor cell got the right mutation, it could just as equally go on to form a tumor. And then the other hypothesis would be that the more mature differentiated cells could also give rise to a tumor. But most people would argue that these specialized cells that are post-mitotic are less likely than a stem cell to do so. But there actually has been very little evidence either way. And for most tumors, we really don't know which cell the tumor starts in at all. So, and most of this kind of work has actually been done in the hematopoietic system, where there is already evidence that stem cells or progenitors could give rise to tumors. Now, in taking this question then to lung cancer, if we return to the mouse model of lung cancer I described for you, we found that the majority of cells in those tumors were positive for SPC, the type 2 marker, and the majority of cells are negative for the Clara cell marker. And that would suggest that perhaps these tumors came from type 2 cells. And this has also been seen in many patient samples of adenocarcinoma. But in contrast, other patient samples are only positive for the Clara cell marker. So both of those cell types were thought to perhaps be the cell of origin of these tumors. But what I did was to go on and take our knowledge of BAS that we had identified and ask, in our mouse model of lung cancer, how might they play a role? And what we found was that in the normal mouse lung, when we stain for this Clara cell marker in red again and SPC in green, you probably can't see it very well here, but we could usually only find 1 or 2 of these cells at the end of a terminal bronchiole. But in the KRAS mice that had been given CRE, we found an increase in the number of these stem cells. And this then suggested to us that these cells might be the cells that go on to give rise to tumors. And this is a time point even before the mice develop tumors, but when we begin to see that hyperplasia, which we think might be the precursor lesion. We also could take KRAS mice and isolate BASCs using the cell sorting method I told you about, or use isolating type 2 cells, and we could activate the mutant form of KRAS by using adeno-CRE in culture or using a control virus. And in this case, only BASC had an increase in cell number after we activated oncogenic RAS, and the type 2 cells did not. So together, these data indicate to us that after insults like oncogenic KRAS or perhaps loss of other tumor suppressors, that we see an increase in the number of BASCs, and that somehow this then leads to the formation of a tumor, which mostly seems to be containing cells that resemble the type 2 cells, but still harbors a set of cells that share the markers of BASCs, the stem cell population we found. So we then also want to, so this then would lead to the idea that BASCs might be the cells of origin of adenocarcinoma, but what about other types of lung cancer? So other groups have shown, as I mentioned, a mouse model of small cell lung cancer, and they have hypothesized that neuroendocrine cells, or perhaps a stem cell that maintains neuroendocrine cells, might give rise to small cell lung cancer. But certainly both of these concepts are still hypotheses that we need to test by developing better tools to ask these questions specifically. And finally, we also want to understand, as I mentioned, the biology of the advanced tumor. And for many years, it's actually been appreciated that many of the cells within a tumor are not equally identical. For example, when tumors are isolated and we get a single cell suspension of tumors, it's actually very difficult to grow them in culture, and only a fraction of the cells in a tumor can be propagated. And that's true in culture as well as in transplantation studies. And this was very intriguing and has actually been observed for many years, and has certainly led to questions about why these tumor cells are not all able to function similarly. And this has then led to 2 different hypotheses. And one would be the stochastic hypothesis that any given tumor cell might acquire the mutations that are needed for it to be able to grow in culture, or to be transplanted, or to form a metastasis, or the cancer stem cell hypothesis, which is a relatively new term. And this idea is that only a small fraction of cells in a tumor that are called the cancer stem cells, that these are the only cells that can propagate the tumor, and that the other cells in the tumor do not have this ability. And this may actually be more related to what I pointed out to you earlier, that cancer cells may be similar to stem cells. Maybe this is important once the tumor is formed. Maybe it's not as important as the cell of origin. These are still open questions. So in identifying a cancer stem cell, what many other groups have been able to do is to take a patient sample, a tumor, and make a single cell suspension of that tumor, and isolate only usually a very rare fraction of cells, and show that these cells alone are able to give rise to what we call a secondary tumor. And this has been shown for leukemias, breast cancer, colon, and brain cancers in patient samples. Now, the important idea behind these cells is that cancer stem cells might be more resistant to drugs than other tumor cells. And that's because when we treat with conventional chemotherapy, we may actually leave behind the cancer stem cells. And because they have this ability to propagate the tumor, or perhaps self-renew, they're able to then give rise to essentially another new tumor, allowing for recurrence of disease. So if we can find better ways to target these cells specifically, we may be able to combine that with conventional therapy and then more effectively treat the disease. So this is very exciting, but we certainly need to know whether every type of cancer has a cancer stem cell and how to define it. And we don't know whether lung cancers have such a cell type or not. So what we've done is to create an assay where we can start with a primary tumor, either from patient samples or mice, and use that same FACS sorting method to isolate different populations of tumor cells. And then what we do is transplant them into nude mice, which are immunocompromised, and so they won't reject the tumor cells. And we've seen that we can get these secondary tumors. And what we're doing now is to transplant different populations. For example, only the SCA1 positive cells, or only the more differentiated cells. And just to point out the more important part of the slide, we're looking here at the percentages of mice that develop those secondary tumors. And when we look at those cells from the tumor that resemble the normal tissue stem cell BASCs, we can see that here, when we inject 1,000 of those cells, about 20% of mice get a secondary tumor. When we inject the other part of the tumor, however, it also gives rise to secondary tumors about 20% of the time. So this actually indicates that this SCA1 marker does not distinguish between the cells that might be a cancer stem cell, or there may be other markers that we need to use. So we're certainly continuing these studies. And so in summary for this part, what we think is that we've uncovered a normal tissue stem cell BASC, and we think that these cells give rise to lung adenocarcinoma, and that alveolar type 2 cells are less likely to do so. This indicates that a normal tissue stem cell might be the cell of origin of a tumor. However, once we think about the concept of cancer stem cells, we've found that both BASC-like and type 2-like cells seem to be able to propagate the disease, but we don't know whether other cell types from the tumor can do so, and we don't even know yet whether there is a cancer stem cell population here in these lung tumors. So these are important concepts for cancer biology and stem cell biology, and they're important to consider probably in parallel. Now, I don't want to overwhelm you, so I'm going to skip through a few of these concepts that I wanted to show you, just some other things that are developing in mouse modeling of lung cancer. But certainly there are examples of using imaging and gene expression, as well as the pathways that are important in lung development seem to be also found mutated in some human lung cancers. So there's a lot of overlap now between lung cancer studies and development and stem cell biology. And as we look to the future, we certainly need to understand how this is related to the human disease, use new technologies to develop ways to screen. We need to develop tools where we can mark only these cells and follow them within the lung and determine if they really are stem cells or if they really are the only cells that can give rise to a tumor. And I think overall, a concept that we can gather is that now that we're understanding that we can use stem cell biology to study lung cancer, we need to put these ideas together so that when we're trying to develop methods of detection, maybe developing a way to detect a stem cell population that has a mutant oncogene, maybe that will help us if we consider these concepts together. And when we look at new potential treatments, we need to think, is this treatment going to target a cancer stem cell, or is it only going to target other cells in the tumor? So hopefully, as we move forward and we study either normal lung stem cell biology or lung cancer, whether it's separately or together, they will inform each other. And so in closing, I just want to thank my lab, which I started less than a year ago at Children's Hospital, and as well as the Harvard Stem Cell Institute. And thank Tyler Jacksware, in whose lab I performed many of the studies that I pointed out to you. So I'll be happy to take your questions.
From what I understand, one of the new hypotheses out there now with your cancer stem cell theory is to find some way to target the cancer stem cell itself because that's what's proliferating the tumor? If you just kill the other cells, then the stem cell will make more and the tumor will come back, is sort of what I understand. My question is, especially after yesterday, the idea of the stem cells being what helps regenerate the lost tissue. So if you're killing off those stem cells, is there a theory that maybe there are some stem cells that regenerate and some stem cells that are cancer causing, or is it one? I guess that's sort of the conundrum. Like, what?
Right.
Can you speak to that a little bit?
Yes. So what you mentioned is certainly the hypothesis in wanting to target these cells. And certainly we think that the stem cells are what maintain a normal tissue. So if we found a drug that could kill, for example, lung stem cells, maybe that would prevent us from repairing our lung after injury. So what we'd like to do is find a way to target only the mutated stem cells that have acquired the mutation that might form the cancer. And that, we don't know how to do that yet. There are a couple of examples in the hematopoietic system where another group has shown that one particular drug can effectively kill the leukemia stem cells, but it actually does not have the same effect on the hematopoietic stem cell. So it's possible that we will be able to uncover those kinds of drugs. So for example, this KRAS mutation, if we can, now that we know about BASCs, and we know that in our mouse model that having this mutation in BASCs, the KRAS mutation in BASCs will lead to their expansion, we can now better compare the properties of those two populations. And one method that I didn't get into is looking at gene expression with microarrays, where we can find genes that are perhaps only expressed in the cancerous stem cells, if you will, and then try to find inhibitors of those particular proteins. That's the concept.
Probably along these lines, I've been reading of certain approaches where they're trying to target the telomerase itself, and kind of join that with other chemotherapies. Is that related? I mean, is that what they're doing there? Because again, it comes to the problem: if you're going to attack the telomerase, what's going to prevent you from hitting normal stem cells? So is it the idea of trying to wed that with certain chemotherapies or certain things that might make them more susceptible?
Right, it's certainly the idea. And actually telomerase is interesting because in a lot of human tissues it's thought that maybe only the stem cells have telomerase, but again, the tumors seem to have activated it. So the fact that stem cells in the normal tissue might share this property with the cancer cell then leads to that whole origin question. But we have to find ways to distinguish them too. And it's certainly a concern that if we have a drug that is effective against a tumor, then we don't want to inhibit the normal function of those cells. In fact, that's why many cancer patients suffer from the side effects of chemotherapy, because it often kills off hematopoietic stem cells, and some of the new drugs are finding better ways to help people keep normal numbers of their blood cells, and that's very important for them to recover. So yes, those are all important issues. Yes.
It sounded like you're making a pretty compelling case. I thought you were going to conclude that you had found the cancer stem cell, which was the BAS cell. And then at the end you said, well, we're not even sure there is one, like you backed off. Is it because of the fact that the AT2 cells, did that really knock you guys sort of, you didn't expect that?
Well, I think so far it's pretty consistent that we think the BAS are the cells that probably, they definitely proliferate and seem to respond very first to what's going to form a tumor later. And I think our evidence still indicates that they are likely to be the cells that drive the tumor formation. But then the question of later, once the tumor is formed, I think it's actually a different question, and it would have been more exciting perhaps for our studies if the BASC also was a cancer stem cell. But I think it's actually a number of people are finding this kind of trend now, and perhaps this is in a mouse tumor, and yet while there are similarities, we still need to understand how it relates to the human disease too.
Yeah, I was taking from it that the BASC cell was the tumor, the cancer stem cell, and then the AT2 cell, which is a more differentiated cell, I think, right? Because the BAS gives rise to that.
Right, that's
Okay.
what we think.
Then that also could cause tumors in culture. Was that what sort of goes against the idea that there is, that you could get the tumor to form from the later cells too?
Right.
Was that that's where part of the problem was in concluding that it was the BAS cell?
Yes, and really with the whole system I've described for you, even though we know the timing of when we start those tumors, what we don't know is what other mutations happened along the way as the tumor could grow. So maybe BAS start the process, but if a type 2 cell that came from a BAS also got another mutation, now it can equally function. And that's actually a danger of certain therapeutics too, that some people that don't agree with the cancer stem cell hypothesis would say, well, you're going to find a way to kill off the stem cell, but then maybe the other cells are going to get another mutation and they can continue to grow the tumor too. And it's still quite early, but we think, if we can catch it early and do both, it would be a better approach.
In the stem cell cancer cell, the genetic makeup of that cell, does it show any differences than you would find in a normal stem cell? Or do they have the same genetic makeup and it's just that one gene gets activated and in the other case it doesn't?
Right, so certainly after the oncogene becomes what we call activated or mutated, it has other proteins downstream of it that carry out those important responses that might make it drive cell proliferation. What we really don't know yet is how putting that mutant gene in the stem cell, how does that affect that cell's ability to function as a stem cell? Does it prevent it from differentiating? Maybe one thing that we think might be likely is the tumors look like type 2 cells because it somehow prevents that cell from going down the bronchiolar differentiation pathway. And some other ideas for cancer treatment are focused on finding drugs that cause this cell to become more differentiated, because it seems like the more aggressive tumors are called poorly differentiated. So that's another concept that people are trying. And in addition to genetics that you're bringing up, also, I don't know if you've gotten this concept, but epigenetics, the non-genetic components, for example, the methods of silencing gene expression with methylation are new concepts that many people in the area are studying that are probably also important changes. For example, a stem cell might normally have certain genes turned off, and that might be another way to uncover the differences between these cells and a tumor cell.
So the gene is methylated in a normal stem cell?
Yes, some of them, yes.
This idea also being carried through with other types of cancer in other parts of the body?
Again, most of the concepts for this studies have come from different types of leukemia. So for example, in leukemia, it seems like the cell, at least in one type of leukemia, the cells that resemble the stem cell, hematopoietic stem cell, are the same ones that can propagate the disease. But now this concept is being carried into other tissues, and we're just now able to isolate stem cells from the brain or perhaps liver and then ask how it relates to cancer. So it's still very new, but now there are many avenues to look at.
I kind of have a question, off that topic, but first I want to say I was really impressed with your strategy for going after, using the fluorescence labeling and getting that particular population of stem cells. It was very impressive.
Thank you. Very lucky as well.
Yeah.
That's part of it too.
My question is the adenocarcinoma. I mean, does that imply a virus involved?
Actually, the adeno portion refers to more of a glandular structure. So any carcinoma that seems to be glandular-like is an adeno. So there's pancreatic adenocarcinoma. And so that's unrelated to the sort of interesting concept that we use adeno to genetically engineer it. But that's unrelated,
okay.
Yes.
How do you sort, briefly, how do you sort, you said the fluorescent activated cell sorting?
Yes.
How does that work?
Okay.
I can say also real quickly if you want to go over it, but We have a resource that one of the teachers made. It's a
Ah.
PowerPoint that goes through the whole
Oh, great.
process that I'll give to all of you after lunch. We'll talk
Okay.
about it briefly, but we do have a really nice
Oh, great.
tutorial
Okay.
that shows the entire process.
Okay, and I apologize for not showing it. Essentially, the idea is that this machine has a number of different lasers to detect each different fluorescent color. And we have our cell suspension that's been incubated with these antibodies. The antibody with this color is bound to our cells of interest. And that tube gets attached to the machine, and a vacuum pulls it in such a way that one cell passes by these lasers at a time. And then the machine will detect whether it has that color or not. And in order to sort the cells out, you can say, okay, the cell that has that color, I'm going to put a charge on it, and that charge will make it go out of a different end of the machine into the tube that you want to collect it into. That's the basics of it. I don't even completely understand it, and I hope that your handout will give you some more detail. But it's actually an amazing technology that I believe came about in the late 80s and is really probably the major tool that allowed stem cell biology to be where it is. So it's certainly a really nice technology to have.
You can do all of that and the cell's still living just as good.
Yes, well,
Wow.
and with gene expression, some people ask, well, does that change it? And the cells are exposed to pressure and being forced through these tubes and lasers, and yet they are still able to grow. So it's quite handy. Yes.
Do you think it's possible for cancer cells, stem cells to be in fetal cells?
So, you mean whether
Or fetal stem cells?
fetal stem cells like?
Would they be subjected to perhaps the same
Okay.
idea?
Yes. Well, I think it is possible. So, for example, it might be that in certain types of childhood cancer, this process of perhaps a stem cell or another cell getting the right mutation might have occurred in a stem cell even during embryonic development. It's very likely for things like retinoblastoma, where it probably started very early in development. And there are actually some types of cancer that only seem to occur more often in children and not in adults. And that's a very intriguing question of whether, does that mean that the cells that form those tumors are only present during a certain stage of development and they die in the adult normally? These are questions that are being pursued too.
Back in 1981, I had a son who had a hepatic carcinoma, and he was diagnosed when he was 9 months old. And I always thought that perhaps that was there during fetal development, and it took them 9 months probably to manifest into the tumor.
Right, and we certainly, in most cases, probably don't know when it occurred, but it is possible. And especially within the liver, it's a very unexplored organ for stem cell biology. So it's hard to know which cells or what mutation occurred in the timing. So, right.
Thank you.
Thank you. And I'll be happy to take any other questions.